everybody. I'm really excited. I think that Pounder Nation will really enjoy this interview today. So today I have the pleasure of hosting Janice Chen. She's the Chief Technology Officer and co-founder of Mammoth Biosciences. Mammoth Biosciences is a biotechnology company based in the San Francisco Bay Area and is powering the next generation of CRISPR products across diagnostics and therapeutics. So Janice, you have quite the resume. You graduated from Johns Hopkins University with a Bachelor of Science in Molecular and Cellular Biology. You ultimately went on to receive your PhD from the lab of Nobel laureate Jennifer Doudna at University of California, Berkeley. Janice is known for her work with CRISPR proteins and is well known for co-inventing the programmable CRISPR-based detection technology Detector. Janice was selected as Forbes 30 Under 30 in healthcare, Business Insider's 30 Under 40 in healthcare, Endpoint's top 20 women in biopharma, and MIT Technology Review 35 innovators under 35. So Janice, I believe it was about a year ago, I watched you deliver a TEDx talk discussing how CRISPR could democratize diagnostics. So it really solidified my belief in the ability to apply CRISPR beyond therapeutics and into other applications as well. And so if any of the listeners haven't seen it yet, I'll attach that link to the video below. And I really highly suggest people go ahead and watch that. So Janice, my first question for you is, can you share with us a little bit about your early career before your time at Berkeley?
Absolutely. And first of all, I just wanted to thank Nat for giving me the opportunity to speak on your show. I remember looking at your first message on Twitter and thinking, wow, this guy's pretty gritty and extremely passionate about CRISPR. So it really reminded me of my early days, sending out cold emails and people who graciously just stepped in and opened doors for me. So now that I can be in a position to give back, I really enjoy helping others achieve their goals. So I just wanted to start with that. I
love that.
So yeah, like Nat said, I'm a co-founder and CTO of Mammoth. We're a biotech company based in the San Francisco Bay Area, developing the Nobel Prize winning CRISPR technology as a programmable search engine for the genome. And so, when I look back at my education, it's really interesting to acknowledge some of the key turning points in my journey, all of which have really involved extraordinary mentors at each step of the way. And I would say my first hook into the life sciences happened when I was an undergrad at Johns Hopkins and took a course called Build a Genome, which had the vision of building the yeast genome from scratch. So this was my first, deep dive into molecular biology, and it really inspired me to study metabolic pathways in yeast, and that eventually brought me to the world of synthetic biology at Harvard. So, in those early days, I learned about the power of metabolic engineering and use some pretty old school genome engineering techniques, which were far less efficient than CRISPR to engineer microbes to produce biofuels. And that experience made me realize just the power of engineering biology, and it inspired me to go back and really understand how these tools work from a mechanistic level and how we can go back and learn to further invent new ones. So in my plan from going to graduate school, I knew that Berkeley really stood out as a university that was deeply committed to basic science, which is why I ultimately selected to go to grad school there. And actually at the time, I only had a surface knowledge of CRISPR. I heard about it and decided to learn more about the science. And that's when I became deeply fascinated by how these, they're frankly molecular machines, these
Right.
Cas proteins, and how they use this RNA molecule to perform just a number of elegant functions to search the genome, interrogate a matching target, and then cut both strands of the DNA. So it was just this really elegant system that was just starting to, gain traction. So I was really fortunate to join Jennifer's lab. And as a grad student, my goal really was to understand the fundamental mechanisms of these CRISPR proteins, which at the time there were just a ton of unknowns, and became totally fascinated by the biology and the mechanism, and just really wanted to contribute by understanding how these enzymes work.
So for your PhD, you then landed a spot in Dowdens Berkeley lab where CRISPR editing had been co-developed in 2012. So could you give us some insight about what it was like working in Jennifer Dowdens' lab and some of the great people you worked with? And are there any key accomplishments during that period that really stick out with you?
Yeah, so I was really fortunate to have the opportunity to join the Dowdens lab at Berkeley, and Jennifer is an incredible mentor and role model, and has always been able to recruit some of the most talented grad students and postdocs to our lab. So it was just amazing to be part of that environment. I think also because of Jennifer's leadership, there was a really strong sense of collaboration, not just within the lab, but also across the entire field and community. And her commitment to curiosity-driven science, I think, was really kind of core to a lot of the students and postdocs there. And actually, a lot of people in the lab have now gone off to start their own companies or their own labs. So it was a really great, environment for a young trainee like myself and others. I think other parts of the lab that really stood out was that the research spanned from discovery of new CRISPR systems all the way to understanding the mechanisms and the applications, right? And so actually in hindsight, it's kind of interesting because the scope of how we thought about CRISPR in the Dowdens lab, from the discoveries to applications, was also a big influence on how we eventually built Mammoth. And the other piece of this is, one of my co-founders, Lucas Harrington, also joined Jennifer's lab at the same time as me, as first-year graduate students. And in a lot of ways, our scientific collaboration and friendship was an important factor for starting a company together.
Right. So you hit on a few topics there. Was there anything you observed during your time there specifically, or was there anything that gave you the confidence to then take a leap and then to go and co-found your own company?
Yeah, so maybe before I go there, I can back up a little bit and talk about some of the things that I'm really proud of, my time in the Dowdens lab. I think first, as a new graduate student, this was back in 2014, the early work in Cas9 had just come out. This was a tool for genome editing and ton of traction in sort of demonstrating how Cas9 could be used in these different gene editing applications. So I was really interested in, okay, can we understand just the fundamental mechanisms of how the protein works? So how does it actually find the target, and then how does it make a cut? And then, collaborated with other labs and using single molecule experiments to study how these individual parts of the protein actually move in relation to binding and cutting sequences. And part of that work basically evolved into being able to engineer Cas9 proteins to be better and more specific at gene editing compared to its natural version. So that was one of the things. Another kind of area that really became impactful to starting a company, of course, was around that time, there was really an explosion of new CRISPR proteins that were discovered. And one of those was the Cas12 enzyme that also made double-stranded breaks in the DNA, but it actually looked very different from Cas9 from both the sequence and structure perspective. And so in trying to understand how this Cas12 enzyme worked, like in the same way we tried to understand how Cas9 works, we kind of stumbled across this unexpected finding that you could actually use Cas12 to not just, cut a target, but actually detect DNA, and that would convert into a reporting event that told you that your target is present. So this was sort of this really eye-opening moment that said, hey, look, you could actually use CRISPR to detect DNA and not just edit DNA. So that was very transformative in our thinking. And then, we ultimately showed you could program the Cas12 protein to detect HPV in patient samples, and that really catalyzed our conviction behind this field of CRISPR diagnostics, which eventually, as you can see, became kind of the starting point for Mammoth. And then there's one last thing I want to touch on, which is that in the time in Jennifer's lab, Lucas Harrington and myself were able to discover, and many others were able to discover, these ultra small Cas proteins called Cas14, which I know we'll talk about later. And we were actually able to show that they are functional nucleases, which at the time was completely against dogma, which is really interesting looking back. I mean, the field really thought that Cas proteins had to have certain structural components, and it had to have a certain size to just work and interact with nucleic acid targets. So it was really phenomenal to be able to show, actually, no, these tiny proteins actually cut DNA, and they also work as a diagnostic tool. So all of these different components, I think, really fed into kind of our belief that we wanted to bring CRISPR beyond the academic lab. And really, we were building the evidence that CRISPR really can be this applications platform. And again, it was that diagnostics discovery which really kind of clicked to us. And then, with our colleague Trevor Martin from Stanford, Jennifer, Lucas, and I decided to just take that leap of faith and start Mammoth. So that was part of our story.
Awesome. I think that's a great intro. And so now, Janice, before we get into the nitty gritty about the science, there's literally an elephant in the room that needs to be addressed. So I think many of the listeners are quite interested in how the team landed on the name Mammoth Bio. So a while back, I read an article stating that several other names are on the table as well. And so do you have any quirky or memorable stories, about when the company was in its earliest days and what the name Mammoth represents?
Yeah, absolutely. So it is a quirky story, and you and others may know that the company was actually originally incorporated as Ophelia Diagnostics, which was a reference to the Greek goddess of simplicity, and it's that simplicity was something that was a feature of Crispr Diagnostics, and that was the link to the name. But we learned pretty quickly that the name was really difficult for people to spell and pronounce. So I don't know if you've tried, but it's challenging. So it was clear that we had to pivot the name. To your point, there's a few things that really inspired us to go with the name Mammoth. So first of all, we were inspired by George Church's mission to use CRISPR to resurrect the woolly mammoth. And as you know, they just recently launched Colossal Biosciences to do just that. So we have no relation to doing that, but it was a true inspiration in the early days. The other piece was that we wanted to pay, homage to Jennifer's other company, Caribou Biosciences. And we liked that mammoth was this large and dominant mammal, also from the Arctic region. So there was a nice little parallel there. And then finally, and almost most importantly, it was easy to pronounce and remember. And honestly, Manny the Mammoth, as an icon and symbol has really taken a life of its own within the company. Yeah. So that's been really fun.
Yeah, I love it. So you co-founded Mammoth in 2017, and just 3 years later, the world experiences a life-changing COVID-19 pandemic where testing becomes commonplace and essentially synonymous with virology and controlling outbreaks. When the rollout of testing began in the spring of 2020, the US Food and Drug Administration gave Mammoth and many other small companies an emergency green light to sell their tests for the virus. But I guess in the early days of the pandemic, you could say we were in a race against time, and that meant that regulations need to be loosened a bit. And so Mammoth ultimately won $30 million in government funding since the pandemic began. So Janice, could you share a little bit about Mammoth's efforts during the pandemic?
Yeah, first of all, I just wanted to acknowledge all the extraordinary work by people in our industry and healthcare for stepping up during, this pandemic emergency, and also the staff at Mammoth for really pushing through significant challenges of operating throughout a pandemic. I mean, it was not trivial in the early days, and even still trying to, work under these conditions. And our story with COVID started in the early days before there was an official name for the virus. I don't know if we all still recall that it was called the novel coronavirus. And this was also before WHO even declared a global pandemic. And at Mammoth, we've been building the foundation for our CRISPR Diagnostics platform called Detector. And I distinctly remember a call with one of our SAB members, Dr. Charles Chu, back in January of 2020, who had some of the earliest novel coronavirus samples from the outbreak in Santa Clara and the outbreak on the cruise ships that had docked in Oakland. And at the time, we had no idea of the extent of what would happen in the following months, but we on the call were just really just concerned about the situation, and decided that Mammoth would reconfigure our detector platform to target the novel coronavirus. And it took about 10 days from designing the assay, receiving the reagents, and then demonstrating that the method works on patient samples. And actually, we ended up publishing our work in Nature Biotechnology in April of 2020, and that was the first peer-reviewed article using CRISPR to detect SARS-CoV-2 on patient samples. And that early work really just catalyzed a number of collaborations with academic, industry, and government partnerships to really accelerate the development of our methods. So, like you said, we received an EUA authorization in the summer of 2020 for our initial method. And since then, we've been working with the NIH RADx initiative to implement and scale that assay onto a high throughput platform. And that's work in collaboration with Millipore Sigma, Hamilton, Agilent, to really help laboratories meet their COVID testing demand. In addition to the work with NIH, we also want to contract with DARPA to integrate these chemistries onto a point of need format towards the vision of using CRISPR for on-demand testing, which we know is an unmet need that's really been exacerbated by the pandemic.
So it's no secret that the diagnostics business is difficult to break into. So there are already a few companies with well-established technologies such as nucleic acid amplification tests like PCR or rapid antigen-based testing that dominate. So we know gold standards like PCR relies on cycling of several different temperatures, specialized thermocycler equipment, but attributes that are unique to Detector include specificity, target flexibility, multiplexing capability. In layman's terms, Mammoth's CRISPR-based detector method can find a specific sequence in a sample, cut it, and unleash a fluorescent signal that will report the result. Could you tell the viewers a little bit about traditional diagnostic testing as the current gold standard for diagnostics? And being that CRISPR originally evolved to detect and snip viral DNA, it seems like virus detection is the first logical place to start. And where else can detector really shine with its high fidelity detection?
Yeah, so I'll take that question in a few parts. So
Yeah.
the first is that, of course, the pandemic has taught us about the entire spectrum of diagnostic testing, and that there are different types of tests that have critical roles for specific use cases. And so we can kind of broadly classify them as centralized and decentralized testing.
Okay.
And then we can also segment further from there. But generally speaking, PCR testing has been the benchmark for molecular diagnostics because of its performance, but it has pretty key limitations in terms of going beyond the centralized labs. And this is due to reagent equipment needs, operator expertise, cost, turnaround time. And then, we look at the other end of the spectrum on decentralized testing. These antigen tests have been really successful in reaching resource limited settings because of low cost and the limited infrastructure required. But I think the big issue with antigen tests is that they suffer in performance as a clinical diagnostic. And that's not to say that they can also be really valuable as a public health tool. And there are some key differences, between the public health tool and a clinical diagnostics. But the other thing is that antigen tests can be really difficult to reconfigure for emerging targets and for multiplex testing. So ultimately, the problem here is that the state-of-the-art diagnostics are just unable to keep pace with disease outbreaks. We saw that, front and center in 2020, and oftentimes the attempts to kind of bridge that gap fail to provide high trust information to at the end of the day, support decision-making at the time and in the right environment. So, so how does CRISPR fit into all this? Well, fundamentally, the value proposition of CRISPR is bringing molecular performance to decentralized settings. And like I said, this is an unmet need that's been really largely sought after in the diagnostics business. And so, CRISPR offers the right balance of cost, reagent, and equipment needs, and the ability to really easily update for new targets, and also detect for multiple targets in a single test. Instead of getting a result in days like you might with your PCR test that you send out, you're getting your result in minutes. And most importantly, I think that, in my opinion, the performance on sensitivity and specificity are kind of those key metrics for a trustworthy decision. And that's what you want at the end of the day. And so the other issue here is that even if all these unmet needs are resolved, there's still a lot of challenges for decentralized testing when it comes to things like consumer awareness. Even with the pandemic, a lot of people are not aware of the full spectrum of diagnostic tools. The other piece is that we still need to be able to connect these test results to a non-healthcare provider setting. And, the final piece of this is the high out-of-pocket costs with some of the tests available. So, we feel like CRISPR is really critical in kind of solving a lot of these problems. I think one of the good things about the pandemic is that it has changed consumer behavior for testing in a positive direction. So, we think about this problem through, all these different perspectives from technology, market, consumer, and in our development. And then you have, another part of your question is around high fidelity detection. What are different ways you can use CRISPR? Of course, there's a huge value in the technology for infectious disease, which is an area that we're focused on currently. But, again, it's a platform method and platform technology that can span well beyond just infectious diseases. And in fact, the high fidelity feature of detector is a really exciting attribute that can be leveraged in a number of diagnostic applications. So what I mean by that is the ability to detect single nucleotide mutations. And right now that's pretty much done exclusively in centralized testing with state-of-the-art methods like sequencing. So even for virus detection, the ability for CRISPR to differentiate single nucleotide differences is really valuable. A really great example of this is to detect, let's say, COVID variants, right? Some of these variants just differ by a single nucleotide in the receptor binding domain. And so you could use CRISPR to detect that. Also influenza strains, influenza is a highly mutating virus. And so you can now be able to detect multiple flu strains that you can't do as quickly with the other methods. And then finally, you can extrapolate that feature for indications beyond infectious diseases. So like you said, oncology or immunology, for some of these diseases, there are really well-defined biomarkers that differ just by a few nucleotides between the healthy gene and then the faulty gene. So instead of relying on sequencing methods, just which are really time and resource intensive. Imagine using CRISPR to get the same information in a faster, cheaper, and more convenient setting.
Right. And Janice, this discussion has me thinking a little bit. I don't know if we covered it. What would the sample type be for a test for, mammoth detector test?
Yeah, so that is completely indication specific. In the case of a respiratory virus, right, that would be, just a nasal swab. So there's a lot of support now that instead of having to use, what was considered the gold standard with the nasopharyngeal swab, we can now just go with a swab that just goes up slightly in the nares. For other things, like let's say you wanted to do some kind of genotyping assay and you wanted a non-invasive sample, you could use something like a saliva swab. So just getting sample from the cheek, and that gives you the genetic sample where you can detect a particular SNP. So it really depends on the indication, but the goal is something that's non-invasive so that you can do it in really convenient settings.
Right. And just to cover, do a lot of these legacy methods or traditional methods use a blood prick or something like that? Is that typically what other methods use?
So yeah, some methods definitely use that for things like looking at cell-free DNA. For instance, blood is a really attractive sample for that because it is relatively non-invasive compared to taking, let's say, like a tissue biopsy. So blood is also a valid sample type for the detector method. It's just completely indication specific.
Okay.
Yeah.
Got it. Awesome. So Mammoth is currently leveraging small but powerful miniature nucleases such as Cas14 and Cas5. I hope I pronounced it correctly.
We actually call it Cas-V, but
Cas-V?
Yeah.
So they're less than half the size of the first generation Cas9 nucleases. When my friends and I discuss these nucleases, we joke about the idiom that big things come in small packages. And so I recently read your paper, Programmed DNA Destruction by Miniature CRISPR Cas14 Enzymes, where I covered how Cas14 detector allows for CRISPR-based detection of single-stranded DNA pathogens, as well as high-fidelity detection of SNPs without the constraint of a PAM sequence. So with Mammoth being a CRISPR, jack of all trades, where you have a protein discovery pipeline that feeds into multiple platforms spanning diagnostics and therapeutics, could you share examples of nucleases you have in the toolkit and how you plan to deploy them across your variety of applications?
Yeah, we love the saying less is more, and it absolutely rings true for these ultra small CRISPR nucleases like Cas14 and Cas-V, particularly in therapeutic applications. But when it comes to our CRISPR toolbox, this is a key differentiator of Mammoth because of our ability to harness the natural diversity of CRISPR enzymes that have evolved over billions of years in nature. And so while some companies start with a single enzyme and then engineer different variants, we've seen greater success in starting with a wide diversity of enzymes at the top of the funnel and then sort of narrowing it from there. And so we kind of take this top down and bottoms up approach so we can select the best enzyme for the right application, and we can engineer it further if necessary. So that's kind of what we think about when it comes to the CRISPR toolbox. The other thing I want to point out is that, we talk about Cas12, Cas13, Cas14, and Cas-V, and these are well, used names now, but they really do represent an entire family of Cas proteins. And what I mean by that is that there's actually hundreds to thousands of variants within each of those families. So if I'm talking about, Cas-V, there's a whole, tree around, new Cas-Vs and different types of Cas-Vs. And truly they all have unique biochemical properties. So, we believe strongly that in our approach, there's really no one size fits all kind of enzyme to enable all the possibilities of CRISPR. And so, having this discovery engine really maximizes our ability to uncover new variants that will open up, ways to improve lives, whether that's in diagnostics or therapeutics, agriculture, biodefense, in a lot of different ways.
Mm-hmm. Awesome. And so your discovery process involves in silico analysis from exclusive metagenomic databases and machine learning algorithms to yield CRISPR systems. Can you share more about like expanding the CRISPR toolbox and advantages of your novel Cas proteins in therapeutic applications?
Yeah, so kind of following up with the previous question, we really and absolutely expect that our protein discovery process will continue to yield more CRISPR systems. I mean, there's just so much of nature that hasn't been explored. And the more we look, frankly, the more that nature surprises and inspires us. So at Mammoth, we do take this discovery first approach, and we use this process called metagenomics. And that allows us to mine for new CRISPR systems that are found in really diverse microbial environment. So, you can go to thermal vents or, national parks or even the oceans and collect samples, do metagenomic sequencing on that. And then you can basically in silico get, you can search for new CRISPR systems, and, these exist as, sequences in a computer. And then we have our discovery pipeline, which kind of goes from taking those sequences and then reconstituting them in a lab. And then we can then start to explore, how do they work? What do they do? What applications can they be used for? And what we find is that, these different Cas nucleases have really differentiated properties. And some of those that, we've talked about, like higher fidelity, smaller sizes, expanded targeting range. So these all, again, if you take a one size fits approach, you miss out on a lot of these unique attributes. But when we have a toolbox, we're able to extrapolate a lot of these things. And then, your next question about how we're thinking about and using the advantages of novel Cas proteins for therapeutics. Well, I think one thing I just want to say is that, we've seen enormous success with these gene editing, the legacy gene editing proteins like Cas9. And, I've worked on Cas9 quite a bit, and seen, again, in companies as well, huge success for therapeutic applications there. But, there are still fundamental limitations to these first generation systems. And some of these range from, again, the fidelity, multiplexing, targeting range, and size. And so to overcome these limitations, this is where we've been able to leverage our discovery pipeline to develop new Cas variants that enhance, these possibilities for genome engineering. So again, going back to the size, this is really, really important. And I think one of the kind of next breakthroughs for being able to deliver CRISPR in vivo. And part of that is, these ultra compact nucleases can be put into delivery vehicles like viral vectors, which are pretty well characterized. And the other, these viral vectors have a really defined payload restriction. So you typically can't fit your first generation, Cas9 system plus guide RNA plus anything else you want to append into these viral vectors. So essentially that's off the table, right? And what these smaller nucleases open up is that, all of a sudden you can fit these Cas nucleases in that viral vector. And now you have, a ton of space to put in additional cargo. So that can be, other domains for other applications. That can be multiple guides for multiplexing. These small proteins are less than half the size of your first generation enzymes. So it really unlocks new possibilities for in vivo delivery. And you open up the ability to put in these kind of what we call, CRISPR plus applications, things that don't depend on cutting the genome, where maybe you're just, modifying the nucleic acid.
Right. And so you actually hit on two topics that I actually want to cover also really quick, Janice. So a lot of people worry about double-stranded breaks. I think I should have asked you a question a little bit before when we were discussing the diagnostics. Are double-stranded breaks an issue when we discuss diagnostics? And what is the difference with diagnostics and therapeutics when we talk about DSBs?
Yeah, so for diagnostics, again, you're taking a sample, and then doing that reaction in vitro or in a test tube. So in that sense, when you do a double-stranded break, you're talking about a non-viable sample because it's, let's say a nasal swab or a saliva swab. So actually cutting the target has no implication whatsoever on that sample. What you care about in the diagnostic application is cutting a reporter molecule that tells you, let's say I found SARS-CoV-2 or I found influenza. So in that context, I think that's really cool too, because, you're not worried about, safety for a patient when you're just taking a non-invasive sample.
Right.
On the therapeutic applications, of course, we care a lot about double-stranded breaks because we know that, if that process isn't done correctly, it can be toxic to cells. And that's where, we've seen a lot of really cool tools that like base editing or prime editing, where you're not actually cutting, both strands of the genome, but you're just modifying specific areas of it. And that, and there's also the world of epigenome editing, where in fact, you're not even cutting DNA, you're just changing, the gene expression of certain genes. And so those are, again, just kind of new areas of using CRISPR that don't depend on double-stranded breaks. However, even though double-stranded breaks, if not repaired properly, can be toxic, we know it's a very viable method for therapeutic applications, especially when it comes to knocking out genomes. One thing that cells do very well in the right context is repairing, those breaks, because it's a signal that says, look, the genome is broken, we got to fix it. And so that's how we get, a lot of these knockout features for using CRISPR as an editing tool. And there's examples of that by other groups in terms of successfully knocking out a faulty gene. So it really depends at the end of the day, what the application is. But I think the beauty of, again, using the technology in different ways is that you can modify it to fit the specific use case.
So we also discussed the benefits of using miniature nucleases for in vivo therapeutics. I wanted to touch on, or maybe ask you, is Mammoth focused on delivery at all, in terms of like the payload or what's carrying the payload, or are you more focused on nucleases?
Yeah, so certainly our bread and butter at Mammoth is the CRISPR technology. So that's looking at the nuclease itself and being able to identify these enzymes that have really unique properties and specific properties for the gene editing application. However, we know that the delivery part is absolutely critical for actually making CRISPR therapies a reality. And so while we aren't necessarily going down to identify and create and discover, at least at this time, new vectors, I think one thing that we're doing is understanding how do these enzymes pair with both viral and non-viral delivery methods, and that's really critical, right? There's, again, different levers that you can tune depending on which ways you deliver the enzyme to the organ, but that's an area that we work with other companies that have really established great platforms for that, and coupling those technologies and creating those synergies.
That's great. And so, Janice, to conclude the Q&A today, I'd like to get an idea of where your head is at on the entire CRISPR space today. And as you know, we're starting to see the first wave of clinical CRISPR trial readouts. And so where do you see the future of CRISPR in the next maybe 5 to 10 years or so? And, maybe your thoughts on that, on the future?
Yeah, I mean, it's such an exciting time to be in CRISPR. We know it's just a transformative technology and will truly reshape how we practice medicine. Like you said, the last decade, if you think about it, CRISPR won the Nobel Prize for the work back in 2012 from Jennifer Emmanuel and colleagues, and hasn't even been 10 years. And in that 9 years, right? It's just been a breakout year already for CRISPR for research in medicine. Honestly, the next decade, I believe, will be shaped by actually bringing these CRISPR products to market in both diagnostics and therapeutics to improve patient lives. And we're seeing really incredible readouts by companies, going through clinical trials. So there's a lot of good data and proof points, supporting that. And it's, the proof will depend on how things go for the next, 5 to 10 years. I think overall, we're seeing a shift in the field, right? CRISPR 1.0, we like to call the legacy enzymes CRISPR 1.0, and that was all about finding a site in the genome, cutting it, and making a crude knockout, right? That's been successful, but that's really kind of the first generation. If you think about what's next, right, we think about CRISPR 2.0 or CRISPR 3.0, it's really expanding the possibilities of the technology as a search engine. And this is by pairing new enzymes with new applications. And of course, this is something that Mammoth believes strongly in and something that we're really pushing forward. Again, specific to what we're doing, today, right, diagnostics really is the foundation of sustainable healthcare when you think about it. And so when it comes to CRISPR diagnostics in particular, in the next 5 to 10 years, we'll see these clinical grade diagnostics reaching patients and consumers where they are, and importantly, giving trustworthy information that will empower people to make informed decisions about their health. I think the other part, which is really interesting, and you're starting to see this as well with digital health companies, is like, we are seeing an evolution towards on-demand services. This has existed for entertainment, for consumer goods, for information, and I believe that healthcare is next, and we're starting to see that movement. So CRISPR will play a really critical role in enabling the on-demand testing because of its ability to go into these settings. So then on the therapeutic side, I think in the next 5 to 10 years, we'll see even more breakthroughs for in vivo delivery and editing with CRISPR. Right now, a lot of that work is really focused on organs like the liver and the eye because of the limitations from delivery. And I think we're going to see, again, those breakthroughs also not compromise safety and efficacy for patients. I mean, that's just absolutely critical. I think the other thing is that we'll see, again, CRISPR being used in indications that there are no cures currently, and this is, examples like cancer and neurodegeneration. I think because of our ability to now think about using these, smaller enzymes and being able to deliver to specific organs, you're going to open up a whole world of, taking these undruggable diseases and now making them accessible. And we'll also have more precise methods for genome surgery. And so I think ultimately we're going to see fatal diseases have a CRISPR cure, and that's really, really huge for patients that are suffering from these diseases.
I love that term you use, the genome surgery. That's the first time that I've heard of that.
That's exactly how we think about it, right? And I think we're going away from the crude, hammer and sledge, and let's make these precise surgical changes.
I love that.
Yeah. And then I guess the last thing I'll say to your question, which I love, just thinking about the future, our mission at Mammoth really is to improve lives by reading and writing the code of life. So we're doing this by harnessing the natural diversity of nature for developing these next generation CRISPR products. And I think what's really cool about what we're doing is we'll continue to discover these new enzymes, and they'll have life-saving properties that really exist all around us in nature. And it's really up to us to uncover and deploy those possibilities.
Awesome, Janice. Thank you so much for letting me grill you, for the past hour or whatever it's been. It was very, I had a lot of fun. It was very exciting. I was very much looking forward to it, and it lived up to the hype for me personally. So I really want to thank you. And Janice, if you want to end, the discussion with any questions for me, go ahead and feel free to ask.
Awesome. Well, thanks so much again for the opportunity. I did have one question because I know, you had brought this up, and it really kind of spoke to sort of my own introduction to CRISPR, right? And so my question for you, and I think your listeners would love to hear this, is just what got you hooked on CRISPR, and what excites you about the future of the technology?
Right. So a lot of my followers probably already know. So it was about 4 or 5, so 4.5 years ago, I took an upper level genetics course. And, we were learning, not just about CRISPR, but gene editing was like the overlying theme. And so we covered things from TALEN to zinc finger and CRISPR. And I think when people think about gene editing, they think that CRISPR, was the first on the scene, but it's not necessarily the case. We had TALEN, we had zinc finger, which were popular at the time, and they still are. But one of the overlying themes of the course was that CRISPR is going to be the future, in terms of, costs, scalability, and feasibility, ease of use. So what I took from the course was, obviously I was going to, I had to learn about it, take the course, pass with good grades, but every day I would go home and I would learn about CRISPR, I would study it, about, everything I could read about it basically. And so I took to the internet. And so there wasn't much. It was still very much early days of CRISPR. And so I took to Twitter eventually, and I found that there was a small community of people who knew about CRISPR. And, nobody in my personal life, my family, my friends, nobody had heard of CRISPR. Everybody, they called me crazy when I was talking about like genome editing. They were like, what are you talking about? Like that doesn't sound right. They were telling me to just move on. Like, why do you keep talking about this? But anyway, slowly over time, there was a large community on Twitter that wanted to learn about CRISPR. The only thing is that it's very intimidating. It's not information that, everybody understands. And so I really believe in trying to make this information more digestible and for people to understand. And so that's a big part of what I'm trying to accomplish. That's why I try to have these founders, CEOs, CTOs on my show, so that we can make this information much more digestible and easy for people to, understand. And, if it's something that interests them, they can always go down the rabbit hole. And that's something that I'm very, very passionate about. And I'm going to continue to do so as long as I have the opportunity.
Awesome. I mean, your passion really stood out. And I know many people are grateful for the work that you do to making this accessible. And again, thanks for giving me the opportunity to talk about some of the stuff we're doing at Mammoth, and hope we can continue to educate people on what CRISPR can do.
Awesome. Thank you, everybody, for listening. And I hope you enjoyed.
Thanks.